After studying this module, you shall be able to know the mechanism of the toxicity of various metals in our body, identify the toxic metals, their toxic effects, and the reason for their toxicity. Learn about the chelating therapy and criteria of a potential chelating drug. Know about the various chelating agents and their action over the metal poisoning. Know the mechanism of cisplatin in the fight against cancer.
Dear students, you are already familiar with the elements in biological systems, which have been classified into essential, non essential, trace, and toxic. Essential elements are absolutely essential and necessary for life process. Trace elements are also necessary for life processes. Non-essential elements are not essential. If they are absent, the other elements may serve the same function. Toxic elements disturb the natural functions of the biological system. In this module, we shall discuss about the toxic elements. Before that, let's define toxicity. Toxicity is the degree to which a substance can damage an organisms. Generally, toxicity of the metals may result from blocking the essential biological functional groups of biomolecules or displacing the essential metal ions from biomolecules or modifying the active conformation of biomolecules. Mercury, cadmium, and lead are three such toxic metals whose effect is very fatal. We shall discuss about the three toxic metals mercury, cadmium, and lead in detail. We shall also understand the role of chelating agents in medicine or chelation therapy. Mostly the treatment of metal poisoning is done by the use of chelating agents. It is hoped that the chelating agent will form soluble, stable, and non toxic complexes which are readily excreted. Many drugs are suspected of acting via chelations. For example, diuretics, disulfiram like tetraethyl thiazuram disulfide, tetracycline and its analogues, platinum, lithium, gold, etc. The three toxic elements like mercury, cadmium, and lead. To begin with, let's study about mercury. The sources of mercury pollution are industrial based minings, which contain, of course, mercury as a trace component, pesticides, coal, lignite, which contains about 100 parts per billion of mercury. It is well known toxic metal which came into limelight after the incidence of Minamata disease in 1953 to 1960 in Japan. 111 cases of mercury poisoning were reported who had eaten mercury contaminated fish from Minamata Bay. Among them, 45 people died. These sea fish were found to be containing 27 to 102 parts per million of mercury in the form of organomercury like methyl mercury. The mercury source was the effluents from a vinyl chloride plant releasing into the Minamata Bay. This was also followed by more tragic report of mercury poisoning from Iraq in 1972, where 450 villagers died after eating wheat, which has been dusted with mercury containing pesticides. These two tragic events boosted the awareness of mercury as pollutant and ultimately resulting its being studied more extensively than any other trace element. Mercury can be toxic by ingestion or inhalation, but the toxicity depends upon the chemical form of mercury. Soluble inorganic mercury salts are highly toxic. In excess, these mercury salts causes corrosion of the intestinal tract, kidney failure, and ultimately death.
Cadmium. The source of cadmium pollution in urban areas are metallurgical plants, cadmium plating, and battery fabricators. Acute cadmium poisoning leads to nausea, salivation, vomiting, diarrhea, abdominal pain. Cadmium deposition tends to be cumulative in the kidney with lower concentrations in the liver. Another characteristic of cadmium poisoning is brittleness of bones. Cadmium occurs in nature in association with zinc minerals. A severe outbreak of chronic cadmium poisoning occurred along the Jinstu River of northwest Japan and was known as itai itai or ouch ouch disease. Over 20-year period, around 100 people died. In 1961, cadmium was found to be the cause. The region was due to a old disused zinc mine and river water which was containing cadmium used for irrigation of rice, and people died after eating the rice. Reason for toxicity. Cadmium is similar to zinc. Therefore, cadmium can displace zinc in many zinc enzymes. Like mercury, cadmium also binds strongly with SH groups of cysteine, residues of enzymes. Example, carbonic anhydrase, dipeptidase, carboxypeptidase, etc. Cadmium, like other toxic metal ions, affects the active conformation of biomolecules due to the strong binding. Lead. Source of pollution. Battery industry is the largest single user of lead, but leaded petrol accounts for more than 20% of total lead consumed per year, and 90% of lead released to atmosphere is from gasoline exhaust. The triethyl lead cation is formed from tetraethyl lead by the dissociation of carbanion. Toxicity of this organometallic cation results from the permeability of membranes, including the very discrimination blood-brain barrier, causing several disorders of central and peripheral nervous system, cramps, paralysis, loss of coordination. One of the characteristic symptoms of lead poisoning is anemia. Reason for toxicity. Like mercury and cadmium, lead inhibits SH enzyme but less strongly. Major biochemical effect of lead is its interference with heme synthesis by inhibiting several of key enzymes, involved in the overall process of hem synthesis.
Chelation therapy. After studying about the three toxic elements, one should be familiar what if they are administered by any human being. Metal poisoning can be treated by detoxification by using chelating agents. Let's now understand the use of chelating agent in medicine and that is in chelation therapy. Mostly the treatment of the metal poisoning is done by the use of chelating agents. It is hoped that the chelating agent will form soluble, stable and non-toxic complexes which are readily excreted. Criteria of a potential chelating drugs are: number 1, it must bind the metal strongly to complete for it with biological ligands and excreting as soluble chelate. Number 2, it should be selective for the metal ion. If it is non-selective, then there will be harmful side effects from the removal of other metals, particularly calcium and zinc from the body. Number 3, chelate must be of low toxicity and not metabolized. That is, it should remain unchanged in biological system. Number 4, it should be capable of penetrating into metal storage sites. Number 5, chelate should be less toxic than the free metal ions. A suitable multidentate ligand that can satisfy all the coordination positions of the metal ion would be ideally suited in the elimination of the metal ion, so that the chelated metal ion cannot bind to any binding sites of enzymes and proteins. Some of the chelating agents used in the chelation therapy are ethylene diamine tetraacetic acid, abbreviated as EDTA, British anti-lebesite, abbreviated as a BAL, D-penicillamine, orin tricarboxylic acid, diuretics, disulfiram like tetraethyl thiazuram disulfide, tetracycline and its analogues, platinum, lithium, gold. Let us study about each of them in detail.
EDTA, ethylene diamine tetraacetic acid. EDTA was synthesized in Germany in 1930 by Munz as a substitute for the expensive imported chemical citric acid used as a calcium sequestering agent in the textile industry. EDTA was patented for this use in 1935 due to the greater demand of chelating agents for the removal of toxic elements and increasing risk of nuclear fission products entering the human body. Pfeiffer, Schwarzbach, and others introduced EDTA to medical research in 1945. The first administration to human beings was in the form of nickel EDTA complex for the treatment of breast cancer, but it remained unchanged and excreted in the urine. Later in 1952, it was effectively used against lead poisoning. Sodium salt of EDTA depletes blood calcium levels and produces hypocalcemic tetany. This danger is minimal if calcium disodium EDTA is used as the chelating agent, and it has now become the material of choice in cases of lead poisoning. Calcium EDTA has the lowest stability constant in comparison to the other metal ions in the body. Hence, these metals readily exchange in vivo to form soluble EDTA complexes that are excreted in the urine. Except in massive doses, it is almost non toxic, and treatment with calcium sodium EDTA results in rapid depletion of lead. Calcium EDTA, used for treatment of lead poisoning, acute iron poisoning, and for the removal of radioactive strontium, other than chelation therapy, in which it is generally administered by intravenous infusion. EDTA has also been used in creams and ointments and in hair dyes. A novel application of EDTA chelate is the use of dicobalt chelate as an antidote in cyanide poisoning. The cyanide ion forms a strong complex with the cobalt in the chelate to form a relatively non-toxic and readily excreble species, British anti-lewisite or 2,3-dimercapto-1-propanol. It was used by British Army during World War II to treat patients poisoned by the gas lewisite. It binds to the enzymes containing SH group, but BAL binds strongly to arsenic and removes it. It is also used for treatment of poisoning caused by mercury, arsenic, gold, etc. D-penicillamine. It is used for urinary excretion of copper in Wilson's disease. Orine tricarboxylic acid. It is effective in the treatment of beryllium poisoning. Drugs appear to involve interaction with metal ions. Many drugs are suspected of acting via chelation. Example, diuretics. These are the drugs that promote the formation of urine. Example, dicarbor acetazolamide. It coordinates with zinc in zinc-containing enzyme carbonic anhydrase and stops the enzymatic activity for catalyzing the reaction, in which water combines with carbon dioxide to form bicarbonate ion. When this reaction is stopped, will be no conversion of carbon dioxide and water to bicarbonate ion, resulting in the formation of more urine. Disulfiram, tetraethyl thiuram disulfide, used in treatment of chronic alcoholism. It inhibits molybdenum containing metalloenzyme aldehydoxydase, so the metabolism of ethanol stops with the formation of acetaldehyde, producing unpleasant symptoms and discouraging further indulgence. Drug inhibits aldehydoxydase, presumably via the soft-soft molybdenum sulfur interaction. Tetracycline and its analogues. Tetracycline and its analogues have a number of sites to form metal chelates of fairly high stability. Certain metal ions stabilize the DNA double helix. Unwinding of helix normally arises from the repulsion between negatively charged phosphate groups. Binding of cations to the phosphate neutralizes the charge and stabilizes the double helix. Magnesium ion is most effective in stabilizing the structure. It is now generally accepted that the action of tetracyclines is directed towards the ribosome of bacterial cells and hence results inhibition of protein synthesis. It seems that the ultimate target of tetracyclines is magnesium ion, which is necessary for the stabilization and function of ribosomes. Platinum. Chemotherapy is one of the main weapons in the fight against cancer. A crucial event happened in 1962 when B. Rosenberg, a physicist who was investigating the effect of electric field using platinum electrodes on the division of cultured bacterial cell, found that field generated between platinum electrodes seemed to prevent the division of bacterial cell without simultaneous inhibition of bacterial growth, which eventually led to the formation of long filamentous cells. Further experiments showed that it was not the electric field, but cis-diamine dichloroplatinum, later known as cisplatin, that were responsible for this effect. These species were formed by tiny amounts of platinum from the inert electrodes reacting with the chloride and ammonia that were present in the electrolytic medium. The subsequent studies, the anti-tumor activity of cisplatin has been studied in tumors induced in animals, and the promising results led to the first clinical trials in 1972. In 1978, it was officially approved as an anti-cancer drug in US. Since 1983, cisplatin has been the drug with the highest turnover in United States. Annual revenues were in excess of 100 million US dollars, and about 30,000 patients per year have regularly been treated successfully. For a long time, this compound has topped the list of most successful patent application granted to American universities, Michigan State University. Although effective against the broad spectrum of tumors, the compound is almost universally used in the treatment of testicular and ovarian cancer, as well as some other types of cancer. The cure rate is approaching 100%, especially for early recognized testicular cancer. The most common side effects of cisplatin therapy include kidney and gastrointestinal problems, including nausea, which may be attributed to the inhibition of enzymes through coordination of the heavy metal platinum to sulfhydryl groups in protein. Accordingly, a treatment with sulfur compounds such as sodium diethyl dithiocarbonate or thiourea and subsequent diuretics may counteract these symptoms. Now it is well established fact that the mechanism of cisplatin is based on its interaction with DNA. The first clue came from the filamentous growth observed by Rosenberg. This growth is caused by inhibiting DNA replication while the RNA synthesis and protein synthesis are relatively unaffected. Studies in vitro as well in vivo indicate that platinum binds with nitrogen 7 of two intrastrand adjacent guanine bases of DNA. This binding changes the active conformation of DNA, leading to inhibiting of DNA replication or cell division. The binding of cisplatin to DNA seriously interferes with the ability of guanine bases to undergo Watson-Crick base pairing. Cisplatin is administered by intravenous injection as an aqueous saline solution. Approximately half the platinum binds to serum proteins and is excreted. The rest is distributed among various tissues. In serum, the drug remains largely as cisplatin, now owing to the relatively high chloride concentration, 0.1 molar. As a neutral molecule, cisplatin diffuses passively across cell membranes into the cytoplasm, where it encounters a substantially lower chloride ion concentration. Hydrolysis produces cationic complexes such as that diffuses to DNA, itself a polyanion. They bind to form cytotoxic lesions. The hydrolysis reactions of cisplatin are an important aspect of biological activity. Reaction is shown as since cis as well as transplatin alter the double helical structure of DNA and its replication, the answer of that question, why only cis is active, is very interesting. The trans isomer is resolved more rapidly than cisplatin. However, the concentration of DNA coordinated trans complex begins to decrease after 6 hours, whereas the cis isomerase then still accumulated in the cell nucleus. Only very little trans compound is still coordinated to DNA after 24 hours. These results indicate that the changes in the DNA structure caused by the trans isomer are sensed differently by the endogenous repair mechanism from those caused by coordination of cisplatin. Recently, it has been found that as the cisplatin binds with DNA, a structure specific recognition protein recognizes the bending of DNA containing cisplatin and binds with the DNA. This protein binds specifically to DNA containing cisplatin and not with trans. Though the mechanism is not clear, but this binding of SSRP may be the reason activity of cisplatin and inactivity of trans. Lithium. Lithium is used in the treatment of the manic phase of manic depressive patients. 1 in 2,000 people in the UK receive such lithium treatment. Manic depressive psychosis involve alternating phases of depression and overexcitement. Very little is known about the mechanism of lithium action. From a general point of view, it is possible that lithium could be interfering with aspects of sodium, potassium, magnesium, calcium metabolism. The existence of diagonal relationship suggests that competition between lithium and magnesium for magnesium ions sites must be considering as a priority. Gold. Gold compound were first used in 1927 for the treatment of rheumatoid arthritis and are still used. The major role of gold compounds in chemotherapy involves the treatment of rheumatoid arthritis with gold compounds such as disodium gold thiomalate myoskricin. The actual role of gold in controlling rheumatoid arthritis is uncertain.
Let us now summarize what we have learned in this module. We have understood what is meant by toxicity. Toxicity is the degree to which a substance can damage an organism. Generally, toxicity of the metals may result from blocking the essential biological functional groups of biomolecules or displacing the essential metal ions from biomolecules or modifying the active conformation of the biomolecules. Mercury, cadmium, and lead are the three such toxic metals whose effect is very fatal. We have studied about these three toxic metals in detail. We also discussed that mostly the treatment of metal poisoning is done by the use of chelating agents. Generally, the chelating agent must form soluble, stable, non toxic complexes which can be readily excreted. Many drugs useful as chelating agent for chelation therapy like ethylene diamine tetraestic acid, diuretics, disulfiram, tetraethyl thiourea disulfide, tetracycline and its analogues, platinum, lithium, gold, etc. have been discussed. So, after studying this module, you are very well familiar with the toxic elements and their effect on biological systems as well as how using the chelation therapy the poisoning can be treated.